Now, there were these really interesting experiments that were done with newborn rats. And they go in and they transect the spinal cord. They basically make the rat paraplegic, can't use its hind legs. But in some of their experiments, they made the cut, and at the same time, they grafted in a wedge of fetal. So a rat pup that was still growing inside its mom, they take it out, they take a wedge of its spinal cord, almost like a piece of carrot, and they put it in the cut space. So it's newborn rat cut, fetal rat wedge. And in a good percentage of those experiments, the fetal spinal cord tissue encouraged the neurons to grow through, and the rats were able to use their feet again. And so the embryonic environment and the molecules that are present in an embryo seem to be very different than what happens after you're born. We don't need to be grafting fetal tissue into spinal cords. We just need to figure out what the signals are, the molecules are, the matrix recognition that allows us to put this sort of magic glue in a spinal cord cut that hopefully will discourage scarring and encourage growth and healing. But we're not at that place yet, but we're working on it.
Yeah, I can imagine. So like you talked about cutting the skin, or that your skin kind of is directional. It grows to strength in one way. And then when it's healing, it probably is just trying to fill the space. It doesn't know which direction to go there.
Exactly.
And then that gets insanely more complex when you're talking about a limb or spinal cord or something like that. The organization of the cells, they need to have a bigger picture, basically, somehow.
It's really cool you said this, because I wasn't going to go there, but the experiment where they put the little wedge of fetal rat spinal cord in the cut, if they aligned the polarity, so anterior, posterior lined up, they got a better result than if they flipped it and we had anterior meeting posterior before it went to anterior. So the orientation of the little piece made a difference. So the cells can tell. They can tell anterior from posterior, right from left, grow this way, don't grow this way, walk this way. So it's really, and what we need to do as scientists is figure out what are those subtle clues. And now with molecular biology, you can take a single cell and you can figure out what genes are on in that single cell. So experiments that we couldn't have imagined 15 years ago, we can now start to tweeze apart and see very subtle differences in groups of cells. What's going to make a heart, what's going to make a liver, what's going to make an eye, what's going to make the forelimb, what's going to make the hind limb. And we can start to say, how are they similar and how are they unique? And how do we turn things on and turn things off? And so in development, we want to understand how they turn things on. We also want to know why things stop and don't overgrow, because that helps us to understand questions about cancer and why a cell population will grow unruly and not differentiate into the structures they normally would. There are these really cool experiments were done a long time ago with a group of cancer cells called teratocarcinoma cells. These cells are odd in that they divide and they differentiate into lots of weird stuff. You might have a ball of cancer cells that might have an eyeball or a tooth or hair growing one way. This is like your worst nightmare of a growth. So there was an experiment where they took teratocarcinoma cells from a black mouse. If they inject those black cells, those teratocarcinoma cells, into a mouse, it'll make cancer. But they took those cells and they injected them into an early embryo of a mouse, into a place called the blastocele, the fluid-filled space in the middle of the early embryo. And they injected the teratocarcinoma cells from the black mouse into a white mouse embryo. And they implanted that embryo back into its mom, and a mouse was born. And the mouse was a mix of white cells and black cells, and it was perfectly normal. So cells that are in an adult environment will make cancer in an embryo environment are re-specified, restrained to participate in normal development. And that's all a question of the signals, the clues, this touching, the rules and regs of an embryo saying, no, we're not going to make a teratocarcinoma cell. You're going to make one of the eyeballs. And they do. So you get a mouse that's got a black eye and a pink eye, patches of black on a normal white mouse background, and the embryonic environment retrained. That's why it's so important for us to study embryos, because the embryonic environment really holds the key to so many of these diseases and understanding them.
So I was listening to a podcast that he was talking about some mechanism that they do this in animals that can regenerate, and they had a way of turning off this communication, he called it. And basically when you do that to an animal that can normally regenerate, like an axolotl, that it won't anymore. And also he said then axolotls basically don't get cancer. And he said if you do this same process of not letting the cells talk to each other, you can also give them cancer that way.
Interesting.
I think the more famous one maybe is he rearranges tadpole faces and then they grow back into.
Right. So, I mean, there's a lot of experiments that have been done where you can, so if I take a human embryo and I split it in two early enough, I can get identical twins. That's easy. We all know people who are identical twins. In a mouse situation, imagine taking an early mouse embryo and then another early mouse embryo and another early mouse embryo, and you sort of smush them all together. So we've got 6 different parents made 3 different embryos, and we smush them all together. So you'd think you get this monster with parts going every which way. If you do it early enough, you get one mouse. It's the normal size. It has cells from all 3 embryos. It has 6 parents. And because its immune system develops after it's developing, it recognizes everything as self. So with basically you can make a mouse with 6 parents. For my PhD thesis work in limb regeneration, I developed an assay that let us look at differences in wrist and elbow and upper arm level blastema cells. So backing up, if you amputate an animal at the wrist, it only can make wrist and hand. That's all it can do. It develops autonomously, true to self. If you amputate it halfway through its radius and ulnar, it's going to make the distal end of the radius and ulnar and the hand carpals and digits. If you amputate it halfway through the humerus, half of the humerus, radius and ulnar, carpals and digits. So there's what we call positional information along the proximal distal axis of the limb and a memory of that position in the cells. We know it's a memory because if the blastema grows big enough, before it's differentiated and made anything, you can actually remove the blastema, you can stick it somewhere else, and it's still going to make a wrist and hand. It's called the distal rule of transformation. It only makes stuff that's distal, and it develops autonomously. It's a long story, but in a nutshell, what I did was I amputated animals at the wrist or the elbow or at the mid-upper arm. And the same time, I also amputated the animal at the mid-thigh level, mid-lower leg. So say I've got an animal that's regenerating a forelimb blastema from the wrist and an upper thigh level blastema from the hind limb. And I wanted to know what would happen if I grafted the wrist level blastema, which is going to make wrist and hand, on the dorsal surface of the hind leg. So once the blastemas had formed, I went in and I removed a piece of skin and epidermis from the wound, from the blastema of the hind limb on the dorsal surface, and I stuck the blastema, the wrist blastema, on top of it, like this. And as the hind limb grew out and regenerated, the wrist blastema rode along. and it made a hand on top of the ankle. So I got a foot and a hand. If I grafted an elbow level blastema on top of the mid thigh, it actually displaced down to the knee, and the elbow joint and the knee joint were together, and I had basically two limbs down the leg. And if it was an upper arm level blastema, didn't displace at all, it just stayed stuck. This was the first time anybody had ascribed a cell recognition pattern, a cell recognition property to wrist, elbow, upper arm level blastemas. So it was a big fat hairy deal. What was even more exciting is at the same time I was doing these experiments, which was in the early 80s, a man looking at frog embryos and frog larvae, young frog larvae, and treating them with something called a teratogen, a monster maker, a chemical that would perturb their growth and development. And the monster maker he was using was vitamin A, retinoic acid. Now, vitamin A is very, very popular these days. It's important in skin care and therapy. Kids that have really bad acne may take retinoids to treat that acne. And if you're female and you're taking retinoids, they want you to be on birth control. So you cannot have a child while you're being exposed to retinoids. What Iqbal Niazi in India discovered is that vitamin A changed the positional pattern and the structures that would form from the wrist level. Say, let's give an example of wrist. And then Dave Stockham and a guy named Malcolm Maiden in England, we explored this extensively. Normally, if you amputate an animal at the wrist, it only makes wrist and hand, and axolotls have 4-digit hands, so I'll do this. If I amputate an animal at the wrist, and 4 days after amputation, I inject that animal with vitamin A, regeneration is delayed, and a funny looking blastema forms from this level. And from this level, in a dose-dependent fashion, so if I use the maximum dose, I can get a scapula, a humerus, radius ulnar, carpals and digits. I get a big extended long limb from the wrist. And if I give a little less retinoic acid, I might get a duplicated radius and ulnar. Give a little less, maybe just extra carpals. It's as though vitamin A is walking the positional information of the wrist blastema all the way back up to the shoulder girdle. So then I repeated my assay, taking blastemas from the wrist or the elbow or the upper arm, all treated with vitamin A, grafting them onto the dorsal surface of the leg, and they don't displace. So vitamin A allowed us to predictably modify positional information, and my assay allowed folks to visualize and see that cell-cell recognition, cell adhesivity, stickiness, and identity go hand in hand, and that forelimbs and hind limbs recognize each other because wrists displace to ankles and elbows displace to knees. So forelimbs and hind limbs play by the same rules, but we still haven't identified the molecules that say, you're a wrist, you're an elbow, you're an upper arm. But we have some good clues, but I think it's going to be so subtle. You know, this is the middle forearm, this is the wrist, this is the elbow, this is the upper arm, this is halfway down the upper arm. Is it going to be sort of a slow spectrum of different molecules up and down the limb? And it probably will be. But a lot of it has to do with cell interactions, cell touching, cell sorting out, cell choices.
Yeah, it seems in some sense insanely complex that, you know, the way that all the cells have to organize themselves, but then also kind of simple. You give these simple signals through just a dose of vitamin A and it can react in a predictable way. I guess it's kind of similar to me if I think about like hormone treatment that people are doing now. It's on the news here, right, with the trans stuff and that. But people take hormones, and the way your body interacts with hormones is incredibly complex. It has all these different effects on it, but it's like, it's a super simple thing for you to do somebody. It's just like, if you want to make them morphologically a certain way, it's just this one thing, and it creates all these changes.
timing has a lot to do with it too. And I'll give you an example. If you have a frog embryo and it's divided a bunch, but it hasn't really started to change or differentiate or do this process called gastrulation, which is when cells move from the outside to the inside. And so if you've got this young frog embryo and you take some cells from what will be the future belly of the frog and you stick it on the top, those cells can actually participate with their neighbors and actually differentiate into neuron. Okay, because they're in the right place at the right time early on. You wait another number of hours, like 8 hours, 10 hours, you take those same cells and you stick them right in that same spot. And they're like, I'm not going to play with you. And they'll actually migrate all the way back through the embryo and find where they should be. And so cells have a developmental potency and a developmental fate. And early on, they may have more potential to do a variety of different things. But then as development proceeds, they become restricted, they become committed, they become determined to just make that one thing. What changes? Those are subtle things that we're looking at to try and figure out, like, what makes a cell determined to be nervous tissue and why 4 hours earlier it doesn't care what it does. And it's all because of cell interactions and the numbers of divisions and its neighbors.